Hello and welcome to Alltech's Milk Quality Series. I'm Dr. Luke Miller, an elite dairy advisor on the Alltech on-farm support team. We all know that mastitis plays a key economic role on dairies. Today we're going to discuss Prototheca mastitis and what you can do for control and prevention on your operation. Prototheca are a colorless algae that are found in a variety of environmental sources and can cause chronic mastitis in cows. Currently, there is no effective treatment for protothecal mastitis, and this can result in significant economic losses for producers. Prototheca are widespread in the environment of dairy cattle and are typically present in wet areas containing decaying plant matter, such as lagoons, manure storage areas, water troughs, river runoff, or runoff from silage and feed lanes. Other internal sources could be milking parlor wash water, teat dip containers, milking machine liners. Infections usually occur when the teat end is exposed to very high numbers of the algae during the intervals between milkings. Although spread through milking is not considered a significant source, new infections may occur if we have poor milking technique and a high percentage of cows or quarters are infected with Prototheca. Most cases of protothecal mastitis are subclinical. Slight changes in milk may be observed, such as discolored or watery milk with flakes or clots. Typically, cows exhibit no severe systemic signs, and the infections will persist and progressively decrease milk production and elevate somatic cell count. Elevated somatic cell counts in a case of protothecal mastitis may be in excess of 1 million cells per milliliter. So how do we go about diagnosing Prototheca mastitis on your dairy? Well, first we want to isolate which quarter we're dealing with, and we can do that through a CMT paddle or using a desktop somatic cell count meter to decide which quarter that we are going to then further sample. After we decide which quarter it's in, we can move on to DHI monthly test for a screening, or we can use fresh and mastitis cow culturing as a diagnostic gold standard. So we found protothecal mastitis on our dairy. What do we do next? Where do we go from here? As far as treatment goes on individual animals, there is no effective treatment. Antimicrobial intramammary therapy is not effective. There have been some anecdotal studies and some research trials on antifungal agents, but they have proved to be very short-lived, and the Prototheca always returns. It's very resolute. Treatment efficacy is something for you to think about when we're looking at a group of cows that have unrelenting high somatic cell count and we can't seem to get any better with them. So then what is our best option is prevention. So the best way to control protothecal mastitis is early detection and immediate culling of all positive cows. The longer they're there, the greater chance we have of spreading protothecal mastitis from animal to animal. Bulk tank milk should be monitored routinely for the presence of Prototheca along with other contagious organisms. Milk from fresh cows and hospital cows should be submitted to a diagnostic lab for pathogen identification. If we find that positive cow, she gets a career change and goes directly to the barbecue. If we can not afford to lose that many cows, or if we have special cows that we wish to hang on to, we can look into protocols that are called test and isolate. When we find a positive, she's moved to a string that is Prototheca cows only. They are milked last before the CIP wash. These cows would be considered to have 2 strikes out of 3. If we find any other problem with those cows, they get the career change and head for the barbecue. One of the best ways that we can work on preventing this and preventing the transfer of protothecal mastitis is through quality milking protocols. So an example of a quality milking protocol is what we're going to run through right now. These Protothecas can be found almost everywhere. We need to keep the environment clean and dry and expose these cows to as few pathogens as possible. I'd like to reiterate a point here, and it applies to several of our environmental pathogens involved in mastitis events. If there is enough of it around, it can become contagious in behavior. This is where the protocol for milking comes in. If we have questionable protocol, that's bad. If we have a good protocol, we can halt the advance of protothecal mastitis through our herd. Things that we want to have in a quality milking protocol: a teat dip, bactericidal in nature. strip to see what type of milk we're dealing with and any abnormalities, a wipe to remove any organic material and left residual, teat dip, and finally a post-dip after milking to ensure that we are preventing as much contamination from the environment as possible. If you would like to hear more about what makes up a quality milking routine, please check out the video from my colleague Jorge Delgado, which is found on Alltech's Dairy On-Farm Support YouTube channel. Thank you again for listening to this video in the Alltech Milk Quality series. I hope that you found this information helpful and can implement some of these techniques on your operation. If you found this video helpful, please check out other videos in our series where we discuss somatic cell count and other mastitis pathogens. Thank you again and have a great day.